So it's my great pleasure to introduce the final two speakers. And the first one is Paula Cannon. And you heard from the end of this morning the sort of development of the incredibly rapid development of CRISPR-Cas9 technologies, right from the discovery through to its ubiquity, or its usefulness in various systems, through then to thinking about the types of applications, and including gene drive in mosquitoes and disease control in the environment. You're going to hear from Paula Cannon today about the work that she's done as a pioneer in trying to apply this type of technology to human diseases, in particular to HIV/AIDS and the treatment of prevention, actually, of stopping the HIV virus in its tracks. And it's interesting in looking back at how rapid this technology has become mainstream. And you heard it a little bit this morning, but there was a casualty, and the casualty was the TALEN technologies, which a number of researchers, and probably many of you in this room, were working with. And it's hard to say that CRISPR-Cas9, I shouldn't say CRISPR-Cas9 is facile, but it is much, much more straightforward and easier to implement than the TALEN technology. So Paula had been actually focused very much on how to adapt the TALEN technology in control of HIV, and then quickly switched, like everybody else did, to the exploitation of CRISPR-Cas9. So Paula is professor of molecular microbiology and immunology at the Keck School at the University of Southern California. Don't feel too badly about bringing her up from South California to Toronto, even though it is getting colder, because she's originally a Liverpudlian, and which isn't known for its climate either, better known for its beetles, I guess. And she's been pioneering, as I mentioned, this work in application of gene editing technologies in modifying, for example, the HIV capsid receptor on human immune cells. So without further ado, I'd like to invite her to the stage to tell us about her work.
Thank you, Jim. Okay, hi everybody. We've all had lunch, all right. As long as I stay awake, I think we're good, yes. So as you just heard, I'm going to try and I guess kind of like take the story full circle and talk to you about how gene editing technologies can actually be used in people to fight diseases. And I also think it's quite fitting that I'm going to be talking about HIV/AIDS, because for those of you who heard the speakers yesterday, you'll know that we sort of finished off with a real powerhouse talk from Anthony Fauci from the NIH about the 35-year commitment now we've had to fighting this disease. Okay, so let's just go through the challenges of what we're up against with HIV. So as you probably all know, HIV causes AIDS because it infects and destroys a key player in the immune system, the so-called CD4 T helper cell. And in addition, the virus also presents other challenges. It has a very high rate of mutation, and that allows it to stay one step ahead of our sort of traditional immune responses, our antibodies, our cytotoxic T cells. The virus also has some other tricks. It's learned how to disable, switch off, degrade, and even exhaust other aspects of the immune system. And so really, it's a very sophisticated pathogen. And then if that wasn't bad enough, the final thing that HIV does is that on occasion, it can enter a cell, integrate into the genome, and basically go to sleep. And this latent HIV, which is present in patients even when they take anti-retroviral drugs for decades, is a problem because the virus can occasionally wake up, and that means that if a patient stops taking their anti-retroviral therapy, the infection can just restart. And I've tried to show what that looks like in this cartoon, which shows the levels of HIV in the blood of a patient on antiretroviral therapy. Antiretroviral drugs are incredibly effective. The viral levels really get down to undetectable levels in a matter of days or weeks. But as you'll see from the cartoon, the moment that people stop taking antiretroviral therapy, the virus comes back. And this is basically why we can't cure people of HIV using drugs. Okay, so what can gene editing bring to HIV treatment and possibly also the effort to find an HIV cure? So there's 3 areas I'm going to talk about today: making cells resistant to HIV, improving the immune responses against HIV, and potentially also disrupting these latent HIV genomes. So first of all, how can we make cells resistant to HIV using gene editing? Well, just as a little primer before we get going, yes, I have thrown CRISPR-Cas into a couple of slides later on in my talk to justify my plain fur here. And I guess everybody by now knows that CRISPR-Cas works because an RNA guide is made to recognize your DNA sequence, and this brings the endonuclease activity of the Cas9 protein to that site to introduce the DNA break. But I've also been in this field for a decade, and we started off, and I have to say they're still my first love, working with zinc finger nucleases and then also with talins, which kind of work in a similar but different way, whereby you have to create an engineered protein that recognizes your DNA sequence, and that brings along a linked endonuclease, which makes the DNA break. So I'm kind of agnostic about these. So I've got zinc finger nucleases, talins, and CRISPR all in the talk. So please nobody develop another technology. Okay, so again, a slide you've probably all seen a lot now over the last 2 days, just showing you that downstream of making the DNA break with whichever nuclease system you use, there are 2 potential repair pathways, and they can be exploited to give you different outcomes. If there is just the DNA break, the non-homologous end-joining pathway can be error-prone, although most of the time it isn't, and that can lead to insertions or deletions that knock out the function of the gene. Or on the right-hand side, if you also introduce a homologous sequence that we call a homology donor, we can fool the homology-directed repair pathways into copying that and thereby editing the gene. So when we first started thinking about actually moving nucleases into the clinic, the obvious thing to do was to go with what was simple and think about applications that relied on just gene knockout. But if you think about it, there's not many genes in the body that knocking them out is going to lead to a therapy. And if you guys have any good ideas, I'm always delighted to hear them. But there was one very strong candidate for this approach, and that's the CCR5 gene. Okay, so CCR5, which is a G protein-coupled receptor, is the major so-called co-receptor that HIV uses to get into cells. HIV needs to bind to both CD4 and a co-receptor like CCR5 to get into the cells that it kills. That would be fine, apart from the fact that about 1% of the population naturally don't express CCR5 because they have 2 copies of an allele called CCR5 delta 32 that makes a truncated and non-functional form of CCR5. And importantly, those CCR5 negative individuals are basically completely normal. So this was nature's way of saying to us, here's a gene that actually you, humans, don't need, but it, HIV, does need. So that became really the poster child for trying to go after a gene knockout approach in a clinical setting. There was one other piece of very compelling information to do this, and this was the case of the Berlin patient. This is the only known case to date of anybody who's actually been cured of HIV. And Timothy, the Berlin patient, was cured because he received a bone marrow transplant, a hematopoietic stem cell transplant, as a treatment for leukemia from a donor who was also CCR5 negative by virtue of having this delta 32 allele. So if you like, if this is Timothy receiving his bone marrow transplant, overnight he went from having his own immune system that was CCR5 positive and therefore HIV susceptible to taking on the immune system of his donor, which was CCR5 negative, unable to support HIV infection. And Tim had this transplant over 8 years ago now and has remained off anti-retroviral drugs since that day and is still HIV-free. So this is why we talk about him as being the only case of an HIV cure. So doing a bone marrow transplant on people just because they have HIV is clearly not feasible. And so instead, what we tried to think about is, can we mimic the therapy he had? In other words, can we take a patient's own cells and make them CCR5 negative using gene editing technologies? So in order to do that, you have, I guess, a choice of cells that you can think about editing. You can go straight for the CD4 T cells, which HIV targets, and you can pull them out of somebody's blood. Or instead, you can also think about going to the hematopoietic stem cells, the stem cells in the bone marrow that give rise to all the components of the immune system, including the CD4 T cells. So what I'm going to do is just give you a little overview of what's been happening in terms of engineering T cells, and then talk about how work from my group, of my collaborators, we've been doing this with hematopoietic stem cells. Okay. So using zinc finger nucleases targeting CCR5 was the first in-man use of targeted nucleases in a clinical trial. And this was done by investigators at the University of Pennsylvania in collaboration with the biotech company Sangamo Biosciences. And this is shown schematically here. T cells were taken out of a patient, treated with the zinc finger nucleases, and then put back into the patient. The sponsors and the investigators have reported on this. They report at many meetings, and they've also published a paper. So I'll just give you kind of like an overview of what they've described. First of all, it's meant to be too difficult to look at. This is just a summary of the information you can get on the 5 trials that are described in the clinical trials database at clinicaltrials.gov. So for those of you who are interested, you can find that. And I also put it in a recent review I wrote. But let me give you kind of like the sort of high-level summary of what these trials have shown. So first of all, in terms of safety, the trials have shown that infusing zinc finger nuclease-treated T cells back into a patient is safe and well tolerated. The patients saw increases in CD4 counts as well as total T cell counts, and CCR5 modified T cells, the genetically modified cells, were shown to persist long-term in the patients. In addition, and more interestingly, was there any evidence of efficacy, any anti-HIV effect? Well, there's a couple of, I think, tantalizing observations. First of all, in one group of patients, 9 patients who were chronically infected, all of them saw long-term CD4 reconstitution, and some of them, to various degrees, showed a decrease in this reservoir of latent HIV, measured as total amount of HIV DNA in the blood. And you'll see for some of the patients, it actually reached a 3-log decrease in this reservoir following the treatment. And then in addition, for some of the patients, after they have received their therapy for a certain amount of time, they're taken off their antiretroviral drugs. And the idea here is that, yeah, HIV will come back, but maybe it will come back slower or lower, or maybe the treatment can allow the patients to actually control their HIV even though they are no longer taking drugs. And in 6 of 9 individuals in one of these trials that stopped antiretroviral therapy, 6 of them were able to control their HIV at significantly lower levels than they had before starting ART. And this is just one individual here where the black dotted line shows you the path of HIV down to sort of almost undetectable levels again. So I think there's something going on there, but like any phase 1 early stage trials, we still have to wait for more patients to be done and for more data to come out. But I think so far, to me, it's looking quite tantalizing. Because of that, I've been involved with a team of people trying to do the next step, if you like, with these sorts of therapies and move the treatment away from the T cells and into the hematopoietic stem cells. Because this, in theory, could give us a treatment that would be longer lasting. Stem cells are forever. And also, it could impact other cells of the immune system, such as monocytes and macrophages, which are also target cells for HIV. So just to summarize what was 5 years of really hard work. We worked with Sangamo Biosciences to optimize methods to disrupt the CCR5 gene in hematopoietic stem cells at sort of a patient scale, 200 million cells. And we found that using electroporation of messenger RNAs expressing the zinc finger nucleases was the best way to do this. We also then had to demonstrate that this treatment didn't actually damage our stem cells, that they could still behave like stem cells. And so we did that by transplanting the hematopoietic stem cells into immune-deficient mice to create what are called humanized mice, and to show that compared to control, the control cells are in gray, the zinc finger treated cells are in blue, there was really no difference between the two groups of animals. And then finally, we asked whether these humanized mice, if we infected them with HIV, whether the zinc finger nuclease treated mice were better able to control their HIV. And indeed, we found that to be the case. Over here down at the bottom, I'm showing just the levels of HIV of blood in a group of mice in black receiving unmodified human hematopoietic stem cells. And in red, you'll see that the mice receiving the zinc finger nuclease treated cells were eventually able to control themselves of HIV. So I suppose we could say we have like little Berlin mice over here. Okay, so this is just a summary of where we are in terms of bringing this hematopoietic stem cell trial to the clinic. And I put this up here because I want to make the point that taking a basic technology and moving it into the clinic takes time. It also takes an awful lot of money. I was fortunate to live in California, apart from the weather, because in California we have a stem cell agency, the Institute of Regenerative Medicine, and they've kicked in almost $20 million to push this project into the clinic, first with 4 years of funding for all the basic preclinical studies we had to do, and now they are actually funding the clinical trials, and the first patients have been enrolled into the trials. So hopefully in a year from now, we'll have some information to share more broadly. Okay, so moving on, I also want to tell you about how we can use gene editing to improve immune responses against HIV. Remember, I told you that HIV disables a lot of these. And there's various things that you can think about doing. We could redirect T cells to specifically target HIV-infected cells in a way that's been done especially in cancer therapies at the moment. I also have a project in the lab looking at improving B-cell function in patients. But the little story I'm going to tell you about today is this last one, about how we're attempting to restore function to intracellular restriction factors. Okay, what are those? Well, intercellular anti-retroviral restriction factors are kind of like a whole group of proteins that have been identified that each sort of target specific stages of the retroviral life cycle, and importantly, some of them can also trigger interferon responses in an antiviral state. So we have all these proteins, but we still get HIV infection. And the reason for that is that HIV has kind of figured out a workaround. Like any successful human pathogen, to some extent, has learned how to fight back against our defenses. And listed here are just some of the examples of how HIV is degrading or blocking the action of these three different restriction factors. So, you heard from Professor James's talk about the issues around making genetically modified mosquitoes. Well, I'll be honest with you, I'm talking about making genetically modified people. And the way I sort of justify it is I say, well, if we had HIV and we didn't have antiretroviral drugs, the human population would crash down to a bottleneck, but we would have resistant individuals who will then become, the next generation. What would they look like? So it's almost like this would be the next move that humans would make. Can the gene therapists get out ahead of the curve and anticipate that and think about actually doing that? Okay, so how do we figure out what the next move could be? Well, the trick we've taken is we've looked at the restriction factors, and we understand that there is species specificity in how they recognize and act against different viruses. So what I'm showing you here is 3 different restriction factors. This is just a cartoon of the sort of genomic locus. And the human forms are all unable to inhibit HIV. However, you can find examples of orthologs in other primate species that are actually still able to counteract HIV. And by doing comparisons, other groups have identified, in some cases, just single amino acids that could be substituted between the human and the primate form to create a form that can now re-recognize HIV. So in order to take advantage of that, we basically are talking about, can we use homologous recombination to edit restriction factors at a single amino acid to restore anti-HIV activity? Or as my graduate students like to say, we're taking people and we're making them into monkeys. So, okay. So one of the things we've done recently is we spent quite a bit of time just trying to develop the tools to do homology-directed repair editing in hematopoietic stem cells. These are very, very challenging cells to work with. This is just a summary of a study that we published last year that identified that the trick to getting this to work was to put the homology donor, the DNA sequence that's going to be copied, into an AAV virus, adeno-associated virus vectors. One of the nice things about AAV is it's like an M&M sort of virus. It comes in many different flavors. Excuse me. And we found that a serotype called AAV6 was especially effective at going into hematopoietic stem cells. Here I just have some data to convince you that that's true. On this side, we're looking at the ability to knock in just minor changes to put in a restriction site. Or over here, I'm showing you that we can also use this to knock in a larger insert, such as GFP. And you'll see here that the frequency of stable, site-specific insertion we're getting at the CCR5 locus in hematopoietic stem cells is really high. It's in excess of 20%. So I think that's clearly a level that should work to be clinically effective. So going back to the actual restriction factors, I'm just going to show you a little piece of data for one of them called tetherin. Tetherin is a cell surface protein that inhibits HIV because as its name suggests, it tethers the virus on the surface of the cell and stops it being released. And we would like to be editing, for example, this mutation, changing the residue at amino acid 45 to an isoleucine. But when you think about doing gene editing, there's really going to be three different outcomes that we can expect. So first of all, of course, one outcome is nothing happens. We make a DNA break and it's perfectly repaired, and so we end up with the wild-type human tetherin. Or we can get the edited version that we would like to get. But unfortunately, the other outcome we can get, because of the possibility of error-prone repair through pathways like NHEJ, is we could actually end up knocking out the tetherin gene. And I kind of think that would be the worst of all worlds. So we had to think about what is a strategy whereby we can promote homologous recombination to make this mutation we want, but minimizing the chances of gene disruption. And we realized that the way to do that was to place our initial DNA break in an adjacent intron where hopefully we could do less damage, but still persuade the homologous recombination to copy our donor sequence into the region in the exon that we wanted to change. A little bit challenging because conventional wisdom really has it that the further away you go from your DNA break, the less efficient the process is likely to be. So, we thought we'd try it anyway. So I think we had a couple of people working on this in the lab. And in order to do it, this is the schematic. So we're just focusing on this part of the tetherin gene here. And here's the exon and the intron. And we basically made a series of CRISPR-Cas9 reagents, because yes, they are simpler when you want to make a bunch of them. We made several at the site and several at a distance in the intron, up to almost 500 base pairs away. When we put just the CRISPR-Cas9s into the cells alone, this is just looking at the level of disruption we get, just to show you that all of these worked kind of approximately the same. Between about 20 and 40% of the alleles could be disrupted. We used both SA and SP Cas9. And then we repeated the experiment with the addition of the homology donor to try and get this T45I substitution. And I have to say, to my surprise, we found that this editing at a distance was actually pretty efficient, even all the way out to almost 500 base pairs. So where we are with this now is clearly we're sort of focusing on using the systems that are out here in the sort of safest places in the intron, and we're putting these cells into humanized mice to see whether or not they can indeed confer an anti-HIV activity. Okay, so the last little story I want to tell you is how we could possibly use gene editing to disrupt latent viral genomes. And here the idea is the same as kind of knocking out a gene, except you consider an integrated HIV genome as the gene that you want to knock out. And again, the idea would be that if we could treat people with these reagents while they were on ART, but then take them off ART, if we've removed the latent reservoir, we could potentially allow them to have a long HIV-free period, hopefully indefinitely. So here you go. I put talins in my talk as well. We made some talins that recognized various highly conserved regions of the HIV genome. This is just showing in cell lines that, with the notable exception of this one, I don't know what happened. Just means the data's real, doesn't it? They all worked very efficiently to suppress HIV replication. And in addition, we also have a cell line model of a latent HIV genome. This is a T cell that contains an integrated HIV that also has GFP that can be reactivated by treatment with TNF-alpha. Then this causes the cells to go green. And we find that if we treat them with the talin, this one is directed to the TATA box and the HIV LTR, we abolish that reactivation. So that's great. We can remove the HIV reservoir from a cell line. Right, that's not going to get us very far, is it? So we realized that what we had to do next was try and sort of really look at this in an in vivo system. So the next thing we did was try to develop an in vivo model of the HIV reservoir based on using humanized mice. So I've talked about humanized mice a couple of times already in my talk, but I don't think I formally introduced you to them. So here's what a humanized mouse is. It's basically an immune-deficient mouse that is injected with human hematopoietic stem cells. And because it's immune-deficient, it basically grows a human immune system in the mouse. And then you can infect the mice with HIV. And this is just showing that the mice have high levels of human CD4 T cells in their blood. Our mice also take their medicines. We can infect them with HIV and give them anti-retroviral drugs that we mix up with their food. And you'll see that the levels of HIV, once we start them on the ART feed, plummet. And of course, if we take them off their ART feed, the levels of HIV come roaring back. So exactly like we see in patients. In addition, just like we see in patients, there are lower levels of these drugs in certain key areas in the mice, in particular in lymph nodes. And if we look at the residual levels of active HIV RNA positive cells, again, we see high levels in the lymph nodes, which are observations that have also been seen in people. And for those of you who heard Dr. Fauci's talk yesterday, he was talking about how his lab had done one of the first demonstrations of that phenomenon. So far, they're looking like our patients. And then finally, what we did was we adapted a method that came from the lab of Jerry Zak at UCLA to see if we could come up with an easy way to quantify the latent reservoir. So what we did was we infected the mice with a strain of HIV that had a tag on the cell surface. It expressed an epitope, HA. And so what happens is if you take, say, the spleen out of these humanized mice or lymph nodes, you have a mixture of cells, and the cells that are actively, productively infected are expressing HA on their surface. And that allows us to then put those cells through a system which will deplete all of the HA-positive cells, and then we end up with a pool down here of cells which have no HA on their surface, but are basically a combination of completely uninfected cells and latently infected cells. And we can't tell the difference because they just look exactly the same. However, if you take this pool of cells and then you treat it with two different treatments, including a stimulation step, any latent virus will get reactivated, and this arm of the experiment will now give you HIV. And we can quantitate that, unstimulated versus stimulated, and we see typically about a 10 to 100-fold increase in the amount of HIV that's produced, and also we can look by in situ hybridization. So that's our kind of poor man's latency reactivation assay. We can use that to ask, where is the latent reservoir in the mice? And we found that in agreement with recent data from studies of patients, the majority of the latent and inducible virus is harbored in cells that contain markers of immune exhaustion, expressing molecules like PD-1. And so in this way, the model does recapitulate what we see in patients. And so then the final thing is, can we take these cells and treat them with our anti-HIV talins and see if the talins are able to reduce this reservoir? And the answer is yes. When we take the cells ex vivo and treat them with the talins, this is a control talin and this is the anti-HIV talin, and we get about an 80% reduction in the latent reservoir. So what's missing? Well, what's missing is the big piece of the puzzle. I haven't shown you, because I haven't been able to do it yet, how we can actually take something like talins or zinc fingers or CRISPRs targeted to the HIV genome and put them into a mouse, a monkey, or a person and have them specifically target those cells that contain latent HIV and demonstrate to you that this might work. This is going to be a long project. All right, so to summarize, hopefully, the first bullet point is the most important. Hopefully I've got you excited that of all the things you've heard about gene editing over the last 2 days, what I like about it is it allows you to use your imagination. It allows you to use information about basic HIV biology that other smart people in other labs have published, and you can rationally design an intervention. You don't have to screen for anything. You don't have to hope something exists. You can rationally design it. I talked to you about how zinc finger nucleases were the first targeted nuclease to go into human trials, disrupting the CCR5 gene, and they're being evaluated using both T cells and hematopoietic stem cells in patients. My group has been working on the technology to combine targeted nucleases with, in this case, AAV6 donors, because this allows us to do high levels of precise gene editing in hematopoietic stem cells, and we're interested in the idea of now using that to edit restriction factors as a way to confer HIV resistance. Nucleases that target the HIV genome can disable the virus, and they could potentially be used to get rid of the latent reservoir, but of course considerable challenges remain about the delivery of such reagents. And then finally, we have a humanized mouse model that recapitulates some of the aspects of the latent HIV reservoir in patients, and so this can be used to evaluate methods aimed at their reduction. And I'll just, okay, there is a slide with thanking everybody, and it's disappeared. I don't know, there's something about this computer, isn't it? It chops off the last slide. Well, gosh, can I remember them? Everybody in my group who's probably watching, and they're all cringing now, everybody in my group, everybody who used to be in my group and escaped, and the many collaborators I have, especially John Zaya and Dave DeGusto at City of Hope, and Mike Holmes and formerly Philip Gregory at Sangamo Biosciences, and thanks for your attention.